Let's see here. Brian is here. Curry, not here. Darby is here. Fisher is not here. Hamilton is online. Hellman is not here. Ashley is here. Crystal's not here. Taylor's online. Chase is online. Rollins is not here. Collins is online. Sheehan is online. And Vargas is here. Not quite 8 yet, so we won't officially get started. For reminders, let's see, this week, I double check, but if this week's lab is the video, obviously we don't need to come into the lab for that. You just watch the video on your own. That's it for announcements. Everything else is pretty much on autopilot now. 2-minute warning, so to speak, of the semester. Do you guys have any questions about anything? Fisher is online. As always, if you're online, don't forget to send me the words. Let's get started. We need to move kind of quickly. I won't say we're behind. We're close to being behind. I guess the first word for attendance, we're just going to say is genotype. So usual things if you're here or watching live, but if you're watching the video later, send me a sentence with the word genotype in it. Now this is where we left off, and I'm gonna not talk too much about it, but I want to give you a really quick example of how this might be on the exam. It'll be very basic math. So again, allele frequencies, right? You have, let's just say it like this. Use your chin dimple, for example. All right, so if I give you a question and says, I don't know, 70% of the people, so 0.7, have the dominant gene for chin dimple, or the dominant allele for chin dimple. And what percent have the recessive, right? And that's going to be just basic math, right? So 0.7 plus what equals 1? The answer is 0.3, right? Same thing here with the genotype. So you might get a question where I say, I don't know, 25% of people are homozygous dominant, so we write that as 0.25. And I say, I don't know, again, 25% of people are heterozygotes. So what percent of the people must be homozygous recessive, right? So 0.25 plus 0.25 plus what equals 1? It's be 0.5. Everybody understand that? It's basic math. I'm not going to do anything complicated. If you get any questions like this, again, it'll be just basic math like that. Yeah, and definitely, and you might not even get a question like that, but definitely understand that P squared means, those like the percentage, if you will, the amount of homozygous dominants. 2PQ is the heterozygotes, and Q squared is the recessive homozygotes. So any questions about that? All right, this next one I'm going to go through really quickly. It might be confusing, but don't worry about it because I'm definitely not going to ask you anything like this on the exam. I just wanted to give you an example of why this is useful, right? So we're going to talk about population genetics and health science, right? So this is something I don't even think was in your textbook. This is from your last textbook. But it really is a great example of why we would need to do this, right? Maybe not you guys because you're not biology majors, but why people in general would need this, right? So we can use the Hardy-Weinberg formula, I think I just showed you, to calculate the percentage of humans with an allele for certain inherited diseases, right? So this is a real life example. There's this disease called PKU. You can read about it. It doesn't really matter. But what we know is that 1 out of every 10,000 babies in the U.S. is born with it. So from that, then we can figure out the other parts, right? Because if we know 1 in 2,000 has this recessive disease, right? That means they're homozygous recessive. Otherwise they wouldn't have the disease. Therefore, we know what Q squared is, right? Because again, 1 out of every 10,000 babies has it. So 1 out of every 10,000 babies is Q squared, so to speak, right? And the way you write 1 out of every 10,000 is 0.0001, right? And then we just do some algebra. And again, I'm not going to expect you to do this in the exam. But when you take the square root of the Q squared, also therefore the square root of the other side, Therefore, you get Q is equal to 0.1. Therefore, we know if we subtract 1, right, 1 minus Q gives us P. Therefore, we know that P is equal to 0.99. And then from there, we can also figure out how many heterozygotes we have. And again, I went through that very quickly because the point isn't for me to have you understand how that works. I could take time and really break it down for you algebraically, but that's not the point. The point I'm trying to make here is this is why that stuff is useful, right? This isn't just some academic exercise to confuse students, right? People actually use this for real reasons. All right. That being said, let's jump into microevolution as a change in the gene pool. So again, this is a lot of the stuff that you already did in lab. A non-evolving population is what we call it. We say it's in genetic equilibrium. So I should hold it that. You do need to know that. Or Hardy-Weinberg equilibrium, right? You should know both of those terms. Something is in Hardy-Weinberg equilibrium or genetic equilibrium. It's the same way of saying the same thing. That means they're not evolving. And really, it's a theoretical situation, right? Because in reality, things are always evolving, even if you don't really notice it. So for that to happen, for example, the gene pool would have to remain constant from generation to generation, right? So the percentages wouldn't change, right? So again, using chin dimples. If 70% of humans have no chin dimples and 30% do have chin dimples, then that means generation after generation after generation, it would never change, right? And that doesn't really happen. Because generation to generation change in the allele frequency is evolution on the smallest scale, we refer to this as microevolution. So again, all the stuff you did in that lab, all the stuff we're about to talk about, it's microevolution, right? We're not talking about new features coming out of a species or a species changing from one species to the next that we talked about earlier in the chapter. We're talking about microevolution. And before we jump into this, there's going to be questions. You need to know these things. You need to know the difference between genetic drift, gene flow, natural selection, which we've talked about, we're going to talk about again, and mutation, right? These are all things you need to know, and also sexual selection. I don't know, but yeah, that's on there. So here we go. Hopefully you already remember this, but what mechanisms can change a gene pool, right? It's all the stuff we're talking about. So again, we've talked a lot about natural selection because that's the big one. That's the one that leads to adaptations that help organisms be a better fit for the environment, but there are other mechanisms, and that's what we're about to talk about. The first one is genetic drift. You need to know what genetic drift is. That is the change in the gene pool. Yeah, it's been missing some words there. So that's a change in the gene pool. Change in the gene pool due to chance. Or you could just really remember, it's just change due to chance. So what would not be an example of that would be the pepper moths and the other lab you guys did, right? Because the light moths are being eaten because they were sticking out. The dark moths were camouflaged, so they were not being eaten. That was not by chance, that was natural selection. Here's a good example. This is from your old textbook. Let's say you have this population of flowers, right? They're either red or they're white. You can see we've got some homozygous dominants here, heterozygous there, homozygous recessive. And in this situation, there is no advantage to being red. There is no advantage to being white. There is no advantage to being homozygous dominant. There is no advantage to being heterozygous. There is no advantage to being homozygous recessive, right? It is what it is. And for whatever reason, right, this is generation one. You can see the frequencies here. We have 70% P and 30% Q, right? So 70% big R, 30% little r. And by chance, these flowers that are boxed up, those are the ones that happen to survive. Those are the ones that happen to reproduce. So therefore, their offspring, by chance, this is what the next generation looks like. You can see that it's about 50-50, or it is exactly 50-50, right? So again, we've got red flowers, we've got white flowers, we've got homozygous dominant, heterozygous, homozygous recessive, right? They're all there. Again, there's no advantage to being any one of these things. By chance, these two flowers are the only ones that survive and reproduce in that generation. So here we go, by the third generation, all the white flowers have been wiped out. As a matter of fact, so have all the white alleles, right? The little r's, all have been wiped out. And again, this is just by chance. This is genetic drift. There was no advantage to being red. There was no advantage to being white. So that's genetic drift. So for the exam, you'll probably get a question where I give you some situation and say, this is how the population changed. What kind of evolution is that? And you'll need to recognize that it's genetic drift when it's by chance. The next word for attendance, since we're talking about, will be flowers. if you're watching the video some other time, you need to send me a sentence with the word flowers. And the rest of you just do what you want to do. So, under genetic drift, there's two other subcategories. So you're obviously going to need to know that these are subcategories of genetic drift. And then you also need to know the difference between them. So let's talk about them. The bottleneck effect. And again, it is an example of genetic drift, but this specifically is a result from a drastic reduction in population size. But it still has to be by chance, right? So again, using the moths as an example, that was kind of I don't want to necessarily say that, but one might argue that it was a drastic reduction in population size, but not necessarily. And again, that wasn't by chance, right? There was an advantage to being dark in that situation. So here, it's a big reduction in population size, and again, it's still by chance. So what happens is the population passes through a bottleneck, which is what we call a severe reduction in population size. And what happens is this decreases genetic variability because some of the alleles are lost forever from the gene pool. So this results in the loss of individual variation, hence adaptability. In other words, bottleneck effect is a bad thing. Here's a good visualization of what I'm talking about. So here we have this bottle, and you can think of these little colored marbles. It might be hard for you to see. You can think of these little colored marbles as alleles, right? So we've got some purple alleles, some green alleles, and some gold alleles, right? They're all there. We got some genetic variation. That's the original population size. It goes through a bottleneck. You lose most of the population. It just so happens that in this situation, those mostly the purple ones that survived, right? You've got one green one that survived, and all the gold ones just didn't make it. Just by chance, right? So there's no advantage to being purple, no advantage to being green, no disadvantage to being gold. It just so happens that that's who survived. You could almost think of it in human terms, even though it hasn't happened. Knock on wood. If we have World War III and have thermonuclear war, most of the human population dies. Whoever's left, right, that would be a bottleneck. And whoever's left will be by chance, right? No one's like immune to nuclear explosions, right? So they either happen to be somewhere safe or they didn't. Anyway, any questions about the bottleneck? Here's an example. I'm not going to ask you about this. I don't remember even if your new textbook talks about it. But yeah, this actually has happened. And cheetahs are a great example because it's actually happened twice that we know of, right? So they've gone through two genetic bottlenecks. The one was in the Ice Age about 10,000 years ago, and then again in the 1800s because farmers were killing them because they were killing their cattle. Anyway, so any questions about the bottleneck effect? All right, so remember, again, the bottleneck effect was a subcategory of genetic drift, which is just a change by chance. And we have one more subcategory of genetic drift, and it's called the founder effect. And in a sense, the founder effect is a lot like the bottleneck. We're still talking about a drastic reduction in population size, sort of, except instead of talking about a bunch of deaths, right? We're not talking about a drastic reduction size in the population because of death. We're talking about it because they have founded a new isolated habitat, right? So the founder effect, it's in the name. It represents genetic drift in a new colony. And you can see there's some examples here. You can read about them. You can even write about them for independent work if you want. Hopefully I get around to grading it. But yeah, again, I'm not going to talk about it. We don't have time. But I'll just talk about one, for example. So if you guys know about American Sign Language, it started in Martha's Vineyard. The reason is probably because Martha's Vineyard, at least did, I don't know if they still do, had a higher population of deaf people. Because by chance, whoever founded Martha's Vineyard, those people, that group of people, had a higher percentage of people who were deaf. I mean, not a lot, but higher than the regular. Yeah, there's a lot of interesting examples. And I do want to point this out too. What I like about the Amish example is, when we think about founder effect, we're usually thinking about, again, like a new colony, right? So maybe something on an island. In this case, we're talking about humans, but it's not always an island, right? You don't have to be isolated necessarily geographically. And the Amish are a great example of that, right? The Amish people are not geographically isolated. They live amongst everybody else. But they are culturally isolated, so that's why they have different genetics than the rest of us, because of the founder effect, because they are isolated. Any questions about the founder effect? So again, let me put some X's through here. Those are just examples. You don't need to memorize any examples. I would give you some hypothetical situation in a test, and say, what is this an example of? And you'll have to recognize it as founder effect or the bottleneck effect or any of these other things I'm about to teach you. So any questions about the founder effect? All right, the next one's really easy. It's in the name. Gene flow. Your book actually calls it migration. It's the same thing, because it's the change in the gene pool due to migration, right? Due to some individuals leaving or some individuals coming in to the population. It is the genetic exchange with another population. That's the most important thing. So again, for the exam, I'll give you an example. Say there was this island of really tall people, I don't know, and there was an island of really short people, and then someone invented boats. So then they started mixing gene pools, and they started evening out. So that'd be just some random hypothetical example, and that's what you would need to know for the exam. Your book does also point out that it may result in a gain or a loss of alleles, right? So it could be helpful, it could be bad. Generally speaking, you want more alleles. So if you gain alleles, that's generally good. If you lose them, it's generally bad. But again, like I was just saying in that hypothetical example, it reduces the genetic differences between populations. So again, for the exam, I would give you some hypothetical situation. And if it's reducing the genetic differences between two populations, if there's migration going on, then you should say, oh yeah, that's gene flow. So any questions about migration/gene flow? Again, to remind you, those are the same things. Just almost every other source in the world calls it gene flow, but your textbook calls it migration. So they're the same thing. All right, once again, we're back to natural selection. Because again, it's only natural selection that leads to adaptive evolution. And adaptive evolution is evolution that results in a better fit between organisms and their environment, right? The other stuff is just by chance. This actually helps, so to speak. Your old textbook gives an example of the blue-footed booby. This is just an example, so everything I tell you, don't feel like you need to take notes. I'm not going to ask you anything about it. But the blue-footed booby has a body and a bill that is streamlined like a torpedo, which helps minimize the friction as it flies, right? So this bird flies and it fishes, right? That's how it gets its food. It's flying around above the water, it sees the fish, it dives in after it. That's how it eats. So therefore, you want a body that's nice and, torpedo-like. It also has a large tail that functions as a brake. That's useful too, because birds cannot breathe underwater, so it doesn't want to get too deep, and that's what the brake is used for. And then also, and this is interesting that your old textbook brought this up because we're going to talk about this later, but they also have bright blue feet. The males have bright blue feet, and the females, that's what they look for in a mate, which is interesting because, again, we're talking about natural selection, but that specific bullet point is actually sexual selection. And we'll talk about that here in a second as well. But again, I'll put a big old X to this because I'll remind you, I'm not going to ask you about the blue-footed booby. Just an example from your old textbook, that's what the male looks like. It's kind of hard to see back there, but yeah, the brighter the blue feet, the better as far as mating is concerned. So let's talk about evolutionary fitness. Because again, we're talking about natural selection, and natural selection leads to evolutionary fitness. So let's discuss it. Here's a term you need to know, relative fitness. As the contribution that an individual makes to the gene pool relative to the contributions of other organisms, other members of the population. So usually you'll hear people say survival of the fittest, and that's what we're talking about, survival of the fittest. I don't like that term, because so again, I've said this a lot, especially in lab. It's not all about survival of the fittest. It doesn't matter if you survive, that's only half the story. If you're not reproducing, it doesn't matter if you survive. Again, using humans as an example, if somebody had some mutation, I don't know, on their Y chromosome, and it made them immune to, I don't know, testicular cancer, but it also made them sterile, then yeah, that's going to be survival of the fittest, right? They'll probably be able to survive more than your average male because they're, again, like I said, in this hypothetical situation, immune to testicular cancer. But if they can't reproduce, then it has nothing to do with natural selection, right? Because they're not going to pass along those genes. So any questions about what relative fitness is? All right. Now, this one's a little bit more complicated. I don't think it's that complicated, but this is usually where I get more wrong answers on the exam, so pay attention here. Let's talk about the 3 general outcomes of natural selection. Generally speaking, when we're talking about a population changing due to natural selection, there's only 3 ways that it can change. In your old textbook, uses a population of mice as an example. And again, this is just an example. So when you're studying, don't feel like you need to memorize these examples of mice. For the exam, I'm going to give you some other examples, some other hypothetical population, some other hypothetical phenotype. But in this situation, we're going to talk about mice, and we're going to talk about how their fur changes over time, because that's the phenotype that we happen to be looking at. There's 3 different types, right? Here's what you need to know. It might be a little bit hard for you to see. I'm going to give you an example of all 3 of these, but here they are. Directional selection, disruptive selection, and stabilizing selection. And the next slide is going to be a visualization of this. But for now, let's just talk about it. Directional selection is kind of in the name. That's when the population shifts towards one extreme or the other. And again, we're going to talk about phenotypes of fur color. And that'll be the example I'll use on the next slide when I can show you. But for humans, if I were to write an exam right now, I'll use humans as an example, I might say the average human right now is 5'6". I think that's true. And then I say, and then in 100 years, the average human is 6 feet. That would be an example of directional selection because it's moving towards an extreme phenotype, right? So there's fewer in the middle, now there's more tall people. Or I could do it the other way and say there's more short people, right? That would be directional selection if it goes one extreme to the other. The next one is disruptive selection, and that leads to a balance between 2 or more contrasting phenotypes. So again, using the same weird hypothetical example, if in 100 years there's fewer middle height people, fewer average people, fewer people who are 5'6", but there's a lot more tall people and a lot more short people, not one or the other, because one or the other would be directional. There's both, right? Both tall and short. That's disruptive selection. So in disruptive selection, you're getting fewer of the average, what used to be the average, and you're getting more of what used to be the extreme. And in this hypothetical situation that I'm talking about, tall people and short people. The opposite of disruptive selection would be stabilizing selection. In stabilizing selection, you get more and more of the middle phenotype and fewer, fewer of the extreme phenotypes. A good real life example of that is birth weight of humans. You can look it up if you want for independent work. There's not much wiggle room for that. Let's say birth height, I don't know. The size, the weight of a babies, it's pretty average. I mean, there are some exceptions, but generally speaking, you can look it up. Because a baby that's too large is a little bit less likely to survive, and also the mother will be less likely to survive. And a baby that's born too small is probably less likely to survive. So again, for humans, there's an average birth weight that doesn't vary much, which is amazing when you think about the different size of people, right? We have really huge people, really tiny people, as far as birth weight is concerned. That's the situation. And again, that's stabilizing selection. So any questions about this slide before I show you the visualization of these things? All right. The next word for attendance is mouse. So again, regular stuff if you hear it online. If you're watching this video later, then send the word mouse in a sentence. Anyway, here's the original population, right? We're talking about fur color. There's the extreme dark ones, there's extreme light ones, but most of them are right there in the middle, right? That's the original population. Very few extreme. Most of them are the gray mouse, right? Very, very much in the middle. Now, directional selection, in this case, this example, it's kind of going this way, right? So we move this way to there's fewer of the gray mice and more of the dark ones. That is directional selection. Of course, whoever made this graph could have done this the other way. The population could have shifted this way instead, and we had, fewer of the middle colored mice, but more of the light colored mice. That would also be directional selection. But just to keep it simple, for whatever reason, you decided to make it go this way. That's directional selection. Again, disruptive selection, like I said earlier, is when we have fewer of the middle phenotype, but more of the extreme phenotypes. So in this case, it looks like all the middle colored mice looks like they just went extinct. and then we had a lot more light ones and a lot more dark ones. And then, of course, the opposite of that is disruptive selection. So here we go. In this situation, we have a lot more of that middle colored phenotype, and this basically got rid of the other phenotypes, right? The really light ones, the really dark ones, they're gone now. So for the exam, again, I'm going to give you some hypothetical situation, and you need to recognize it as directional, disruptive, or stabilizing. And I don't ever mean for it to be hard. I always think that this should be easy, but historically, I know a lot of people use these questions. So are there any questions about these concepts before we move forward? All right, like I said earlier, when we talked about the blue-footed booby, that was an interesting example because not only was that an example of natural selection, but it was also an example of sexual selection. So let's talk about that very quickly. Sexual selection is when individuals with certain traits are more likely than others to obtain mates. It's not necessarily about survival, though a lot of times it is inadvertently or indirectly linked. But it's all about sex, right? So again, not necessarily survival of the fittest, because that's only half of it. The other half is you have to mate. So there's two basic types of sexual selection. These are all really common. You should know about them, even if you're not biologists. You've probably heard examples of these your whole life. There's intra-sexual competition and intersexual competition. So intra-sexual competition is competition between the same sex. So for example, males, right? So if you've seen those big horn sheep, button heads, right? Because they're fighting over territory, because that's how they get mates. Intersexual selection of mates, again, the blue-footed booby, right? So that's when the female's choosing the male, usually, not always that way, but usually it's the female choosing the male, and they're looking for certain aspects. So make sure you know the difference between those two. And again, like I said, with sexual selection, it's not necessarily about survival, but it is a little bit linked, right? So obviously the stronger in these hypothetical situations I've discussed, the stronger of the male is probably more likely to win, right? So the healthier male, more likely to win, therefore more likely to pass along the genes. Therefore, in a sense, right, it is a little bit like natural selection, right? Because the stronger won. Or in the intersexual selection of mates. Again, with the blue-footed booby. Healthy male is going to have really bright feet. If it's not healthy, it's probably going to have dull-colored feet. So even though the female is not looking at him like, oh, that looks like a healthy mate, that's basically what's happening, right? It's choosing something that's healthy. Another term you need to know is sexual dimorphism. And that is the distinction in appearance between males and females. It is not directly associated with reproduction or survival. Again, birds are a great example for that, because usually the females are really dull colored and the males are really brightly colored, because it's usually the females who are choosing the males. Which makes sense too. Think about this evolutionarily, right? Males just produce a lot of sperm, and in theory, just put out sperm all day, right? In theory, even a human could probably impregnate a lot of people in one day, a male. Female, on the other hand, right? You have your egg once a month, and then you're pregnant, right, for about 9 months, so you're out of commission there. And then usually, and I'm talking more about the animal kingdom and less about humans, but then usually it's the female who's taking care of the offspring, right? So again, the male in the animal kingdom can often just impregnate the female and then go about their business. But the female, again, takes care of the young. So all this should make sense. Anyway, if you download this PowerPoint, you can click on that link. There's a little video you can watch about evolution there. But are there any questions about sexual selection or sexual dimorphism? All right. In some species, the secondary sex structures are used to compete with members of the same sex. And that's what we talked about earlier, that intra-sexual selection. We're coming back to that. A lot of times it involves combat, not combat like humans shooting each other. And I'll show you a picture on the next slide as an example. And very often it's ritualized displays. So you'll see birds, I love those nature shows with these like tropical birds of paradise. And they have these really, really intricate things. Right? They have to dance the right way, and they've got to build a little nest just the right way and decorate it for the female. Really interesting stuff. Anyway, so this is kind of what I was saying in the last slide, but just expanding on it. So in this one, in this bullet point, we're talking about intra-sexual selection. And in this bullet point, we're talking about intersexual selection. And it is interesting too, because again, I want to point out it's not necessarily about survival. And the peacock is a great example of that. Because that tail helps it get mates, it helps it have sex. But if you think about survival, it's really not good. It's kind of hard to hide from a tiger when you have a big old tail like that. It's even harder to get away from a tiger when you have a big old tail like that. So any questions about this slide? All right, next slide, just some examples, some visual examples of what we're talking about. Sexual dimorphism, you can see it on the left. Colors kind of stink, but if you're online, you can probably see it better. But here's the female, kind of a dull gray color. There's the male, brightly colored. So that, again, that's an example of intersexual selection. And then here we have intrasexual selection. There's two males competing for territory, therefore competing for mates. Any questions about that? All right, so from your old textbook, not this new one, they end every chapter with something called the evolution connection. So you can read about this on the PowerPoints. Obviously, it's not going to be on your slide. I'm not going to ask any questions about it, but this is important for your everyday life. How many of you have, I guess you can't, you don't have to answer this because technically this is HIPAA. So there are patient privacy things, but you can answer it if you want. I'll answer it. I've had antibiotics before, and they always tell you, make sure you take all the antibiotics, right? So you have like 10 days worth of antibiotics. And it's important because a lot of times people will take the antibiotics and then they feel better, like, oh, I feel good, and they stop taking it. But what happened is, once you start taking those antibiotics, you kill most of the bad bacteria, right? Most of them. So that's why you feel better, because you got rid of most of that infection. But there were some tough ones that are left over that would require the full round of antibiotics before you kill them. So what's happened instead is you killed all the weak ones, you stopped taking the antibiotics, and now you've got this new population of bacteria in you, and now they're immune to the antibiotic because you already exposed them to it. You killed all the weak ones. Anyway, last word for attendance. We're having kind of an early day, 15 minutes early. 20, yeah. Anyway, last word for attendance will be soccer. Playing soccer. Yeah, that's it for today. So we'll start back on Wednesday with a new chapter.